Welcome to the Cancer Neuroscience Symposium Conversation Series. I'm Monty Montano, Executive Editor of Wiley's Advanced Portfolio. We're excited to partner with MD Anderson and host this series to hear from key speakers featured in the February 2026 Cancer Neuroscience Symposium. Thank you for tuning in as we explore ideas to advance cancer neuroscience together. We're delighted to chat with Vera and Andreas today, and we have special guests, Moran and Kenzie as well, so this is fun. We like to start with learning a little bit about, these speakers, you, Vera, and Andreas. So, I know that you live in a beautiful city, Heidelberg, one of the oldest university towns, I think, in Europe. Maybe say a few words about how you arrived there and why you stayed.
Very good question. Thank you for that. So actually, I'm already quite a while in Heidelberg because it is indeed such a beautiful city. And I already did my studies here. I think I wanted to come here for studying already when I was a kid. I went here with my parents, saw the castle and everything, and was just like, I'm going to study here. And I did. And then also during my studies, I actually already started working in the lab of Andreas Trumpp. And I was initially planning on going away abroad for my PhD. However, I have to say he managed such a great atmosphere here. It was my favorite internship and master thesis that I did basically. And then I decided to stay because it's great opportunities, great people, and I love the science.
And you mentioned the castle. I did hike that castle. It's beautiful.
Very beautiful. Yeah, you have to go the steps up, but it's worth it.
And Andreas?
Yeah, it's a beautiful town. I did my PhD here at EMBL in Heidelberg, the European Molecular Biology Laboratories, and then moved to San Francisco. And after 6 years being there at UCSF with J. Michael Bishop, I then moved and started my lab in Lausanne at Lake Geneva in Switzerland, and actually wanted to stay there. But then the German Cancer Research Center called me and said, well, we would like to recruit you back to Heidelberg. And that came together with the possibility to start a new institute called HiStem, which is a stem cell and cancer institute. And that actually prompted me to go back to Heidelberg. And I think it was a wonderful decision. It's a wonderful town. It's full of students. It's the youngest town or city in Germany, actually. We have 45,000 students, 140,000 inhabitants, and there's a great atmosphere and a great academic power of different institutions, including university, the German Cancer Research Center, EMBL, and many others.
It definitely drove me to stay here for a while.
Yeah, it sounds like quite the hotbed of productivity and creative thinking. And I mean, I guess it in part explains why you guys have been so incredibly productive and innovative. And I mean, there's this talk of heterogeneity in the tumor microenvironment, that there's a lot there and a lot's happening, and it's hard to study. I think you guys have been pretty much leaders in sort of developing technologies to break down this and deconvolute the heterogeneity down to the single neuron tumor interactions. So I know that there's this cool technique you guys have developed, trace and seek, I believe. Maybe you could just chat a little bit about it and tell us why it's so powerful.
I'm very happy to do that, of course, because I just love the technique as we invented it here. So honestly, for me, it was the step that when I joined for the PhD, I just knew that I would work on pancreatic cancer, but not specifically on what. And of course, for me in the beginning, it was a bit, will it be resistant mechanisms or something like that? But I also felt that the field is a bit overcrowded already, so I was looking a bit more into different directions, and of course, then realized from a pathologist angle, of course, we know that hyperinnervation is there as part of the ecosystem of pancreatic cancer. But I was so confused that no one really studied these neurons in detail. And that is, of course, because of the reason that the cell bodies of these neurons are actually not located in the tumor itself, but actually in corresponding ganglia outside of the tumor mass. And therefore, when we look at any single cell data set, whether it's PDAC or also any other tumor entity, we completely lack this cell type. And so the idea was to change that and include this because we do know, of course, every cell type matters in the tumor microenvironment. We see that for the fibroblasts, for the immune cells, for basically every player there. So we wanted to integrate this and develop that technique where we retrogradely trace these axon structures from the tumor back to the corresponding ganglia where the cell bodies lie, and can actually now did this full technique where we can harvest these ganglia, dissect them, dissociate them, and FACS sort them to actually also sequence them to actually analyze their transcriptomic profile.
I just wonder, as you're talking about it, sort of these synapses between the neurons and the pancreatic cancer. How dynamic are these? Do they, once they're fixed, are they sort of spatio-temporally sort of in place and will stay there, or is there a sort of a dynamic aspect to that?
I would say it's very dynamic. Of course, this wasn't the main focus of the tracing. A lot of people are, of course,
Right.
luckily also in that field analyzing this. And there are, of course, already a couple of papers that also show this beautifully how actually cancer cells and so other cell types can form these synapses.
Mm-hmm.
So I would say it's a very dynamic process. So, I mean, also showing that these axons are still constantly growing themselves in the tumor microenvironment really shows that it has contacts with a lot of different cells within that ecosystem. And then, of course,
Yeah.
reacting to every cell type differently.
Yeah, that's what I was thinking. As the tumor maybe responds to treatment or as the tumor grows, the synaptic network may in fact be, there might actually be spatial communication
Absolutely.
that you can capture. Um,
Absolutely. Yeah. And I mean, one axon has, of course, even contact with a lot of different cells, not just the one
mhm,
cell, but as it's
mhm,
growing like a branch into the tumor, it has, of course, usually contact with, at the same time, cancer cells, fibroblasts, immune cells,
mhm,
all of the different
mhm.
cells. So this is
It's
really
also very,
fascinating.
it's also very interesting the idea that there's cross communication between the cancer and the neurons, and that involves a certain amount of reprogramming. And I know that the trace and seq, I believe, is primarily a transcriptional signature of that reprogramming. But my guess is you're also looking at protein interactions, potentially extracellular vesicles, small molecules, more of an interactome. Can you sort of speak to how trace and seq is sort of adding to the arsenal of tools for understanding the network communications that happen in this interactome?
Of course, a lot of different techniques are necessary to cover everything, and you're right. So the trace and seq itself and what we've published so far only look directly at the transcriptomic profile. Of course, transcriptomic profile not always correlates directly with also what is translated, especially also find a lot of microRNAs deregulated, for example. So definitely, I think we need to combine that with a lot of other data sets that also now other people, of course, again, also looking at this is very nice thing of this emerging field, looking at also spatial transcriptomics, spatial proteomics. We're now also going beyond and using the trace and seq, not for actually trace and sequencing, but also using now single cell proteomics that is established here in Heidelberg. And so to really correlate also, okay, what is the transcriptional profile in combination to what is actually translated? So what is actually also sent to the tumor microenvironment?
Super, super interesting. I'm just thinking about the transcriptional reprogramming and how that might be laid in through epigenetic reprogramming or some sort of uh, almost sort of similar to trained immunity, where you'll have a particular phenotype that's sort of fixed into the cell. Is there any kind of, could you speak to whether it's sort of an acute reprogramming or a longer term reprogramming, and how that reprogramming is potentially placed molecularly in that communication?
This is again a very good question. Of course, I think this is not just a direct change that goes away in the moment that the neuron has no contact with tumor cells anymore. We were actually able in one of these experiments to really show that when we resected the primary tumor, that even, I mean, these were just in mice experiments, yet another month later after we resected the tumors, the neurons still, at least partially, maintain that cancer nerve state.
Oh
Of
wow, it's
course,
like a
here
memory.
yeah, it kind of memorized these changes. And of course, we need to study that way more. But we also then saw that they maintained a bit of this nerve state. And of course, then these cells constantly signal to the remaining microenvironment in that sense. And we were also able to show that when we then re-injected new tumor cells, that they grew out much faster. So of course, this can be due to a lot of things, like of course also fibroblasts were already activated in these tissues and endothelial cells and everything, but
Hmm.
we really could show that part of that is due to neurons, because when we ablated the nervous system, this faster outgrowth was also inhibited, so really showing that it can also foster a bit into the relapse. So therefore also targeting neurons might also be important, not just for direct treatment, but also to inhibit or prohibit possible relapses.
But Dr. Trump, we just heard a lot about the reprogramming of the neurons. Uh, my understanding, and those are the like one of the most mature cells in the human body. You're of course an expert in stemness. How often do we see in mammalians that cells that are fully differentiated undergoing such an extensive transcription or reprogramming in any context, not necessarily cancer? And how hard it is to do that, because in the lab to de-differentiate or re-differentiate, it's pretty, an extensive procedure. And I was wondering, how often do we see this physiologically?
Mhm. I mean, you can of course see it with the Yamanaka protocol, where differentiated cells can be reprogrammed into pluripotent cells. However, that's with inclusion of transcription factor changes. In the blood system, you can actually see some reprogramming or lineage switch, from macrophages towards B cells and vice versa. So there are possibilities that actually this happens. However, it's actually quite rare, as you say. I mean, the reprogramming doesn't turn a terminal neuron into a proliferating cell. We have actually shown that indeed the reprogramming does not lead to an increase in neurons, which then increase the number of neurons in the cancer, but it actually acts on the sprouting. So actually, you have more contact of each individual neuron to, instead of X number of cells, 10X number of cells. And these cells now include not only interaction of nerve endings with tumor cells, but also with fibroblasts, with cancer-associated fibroblasts, with probably immune cells, with endothelial cells, or with other cell types which you find in the tumor.
Yeah, I remember, I had a fascinating discussion with Dr. Ishia Fiedler. He was one of my mentors. And when I presented my project to him, I was talking about neurogenesis. And then he told me, Moran, are you sure that this is genesis of new neurons? Because it's probably not proliferating. And this is when we started looking at axonogenesis, neurotogenesis, what you just explained is like the branching and the growth of the filaments, which is a very different process. But I think, I agree. What we're seeing is that those nerves in the tumor, rather than being fully de-differentiated, they're kind of like transforming or transdifferentiating or acquiring new features, and that makes them more kind of like um, supportive of the tumor growth. And I was always curious, if cancer can hijack those nerves to its own, purposes, to its own benefit, how can we hijack it back? Have you ever thought about, about that in the context, not necessarily the therapeutic, even in dishes, how you reverse this process? Maybe a
Well,
lesson learned from
that-
blood cells.
that's a very good point, and actually, it's exactly what you say. And you see the state switches, you see identity switches, and actually, these identity switches are related to the role the neurons actually play in the cancer cells. So the cancer cells actually select specifically, can actually reprogram, actually induce plasticity, but then at the end, the ones which actually grow, which promote the tumor growth most, will be selected at the end. And those will actually then interact. So it's probably a Darwinistic process where you induce plasticity plus selection of the ones which work best. Well, it's probably quite complex, and it's probably a combination of factors which are not only derived from the tumor cells, but the tumor cells influences also the microenvironment outside the nerve cells, and all these together actually lead to nerve reprogramming. Well, Well, you see also, for example, in cancer in response to therapy, that's a good example. For example, neuroendocrine differentiation in response to escape to anti-hormonal therapy in prostate or breast cancer. So the cells then become more plastic and then differentiate into areas, into states where actually the inhibitor, the chemotherapy or actually the anti-hormone therapy actually is irrelevant. And so this is typically done by specific activation of repressors as well as specific transcription factors, such for neuroendocrine development, for example, of NeuroD1 or others, which are master transcription factors which lead to complete fate changes. I, I, I fully agree with you, Moran, and I think all the known induced and natural reprogramming mechanisms are involving major type of transcription factor changes. And I think that this probably is also the case in neurons, although we are currently looking for evidence for this to actually identify the transcription factors which completely drive, the necessary thousands of changes which are necessary to actually reprogram a nerve. So it's the transcription factor and combination of transcription factors which do that. And the growth factors are sort of helping to induce or select the right ones.
Yeah.
And do you
Can
think
I?
it's the, go ahead.
Oh, no, go ahead, Kenzie.
I was going to ask if you think it's the same factors that are driving how denervation impacts the response to immunotherapy.
Well, the immunotherapy, what we have seen is that if we actively denervate, we actually see that calves do change. They lose their tumor suppressive activity and turn into a pro-inflammatory type of cell, which leads to the influx of blood cells, very likely also T cells, and therefore turn a cold PDAC, so pancreatic cancer tumor, into a at least warm one, which is then again targetable by checkpoint inhibition. And that's something we are currently trying in a early clinical trial we are setting up, where we specifically use use therapies which with a specific type of MR-Linac system to destroy celial ganglion, which actually then leads to denervation. We combine this with nab-paclitaxel, which we showed in the paper and in the mouse model to be already toxic for the peripheral nervous system, but this alone is not sufficient. So a combination of celial ganglion destruction or blockade combined with nab-paclitaxel should actually also lead in the human to the influx of immune cells, and so we combine this even with plus minus checkpoint inhibition. And this is a trial we are currently planning, a multi-center trial in Germany, and we are looking forward to hopefully start this in 2026.
I'm curious about this denervation approach, and does the denervation affect the functionality of other adjacent neurons? I mean, or is it more specific to that synapse that you've just denoted? I mean, is there sort of a feed forward effect or a proximity effect?
Yes, so we were a little bit worried about, by taking block the celiac ganglion, that the side effect might be very strong. However, there is a recent paper which has been published for end stage cancer patients, where the goal is to actually reduce pain. So this approach was quite successful, and so we with reasonable side effects. So we are currently looking forward to actually use this type of therapy.
Super interesting. Just sort of thinking about some of the things you were saying, you and Vera were saying, and I guess, I mean, neurons in the pancreas communicate in normal organ crosstalk, right? And the neurons also communicate with immune cells in normal sort of organ homeostasis. And with the PDAC, and I guess more all of these peripherally innervated cancers, there's sort of an aberration, I guess, of that normal crosstalk. But what I'm still not so sure I understand is what is the aberration? Is it a shift in the kinds of factors that are produced, growth factors, inflammatory chemoattractants? What is the nature of the hijacking? Is it more of the same, only selective targeting of sort of neuronal function and neuronal modulation of immune cells, or is it a radically different kind of functionality that these neurons acquire when they're being instructed by the tumors?
Or it's probably both. So I mean,
All
when
of the above.
right. I mean, in the case of denervation, I mean, we have the most extreme effects, and there we do actually see that the calves actually the calves are reprogramming by actually the absence of neuronal input, which we can then use to actually do targeting using checkpoint inhibition. And so what we're currently doing is this type of radiotherapy, which is non-invasive to block that. That's of course a very crude way to do it. We are currently looking at other targets which we may use in a more specific manner to indeed reverse what Moran was saying, basically reverse the reprogramming, turn cells back into their origin, but also block their activity, which drives tumorigenesis forward.
It's
Also,
interesting.
maybe.
Sorry, go ahead.
Maybe Vera can add to that.
Yeah, so I mean, beautifully said already by Andreas. And coming also a bit back to, in general, the crosstalk between immune cells and the neurons. Of course, it is very hard to specifically link it to just specific targets, because we've seen now specific way of reprogramming in pancreatic cancer, where it's mostly going through the cuff reprogramming. So what we could also do to just in the mouse setting, of course, is directly linking ligand receptor expressions, and then seeing, for example, in the calves, a lot of interleukin 6 was, for example, deregulated then on both sides similarly. So you can bring it a bit down to targets. But what I find very interesting is that we are, of course, now expanding this to other organs in their normal homeostasis, but also to different cancer entities. And so, for example, when we now look at colorectal cancer, the fact we also find a lot of immune modulators, but it's not necessarily the same one, so they differ, which is specifically interesting because a lot of the neurons that innervate the pancreas, or then also pancreatic cancer, are actually the same ones that also innervate, for example, several parts of the colon. So one neuron can actually innervate different organs, and then it's so interesting that one axon might sense a different input from the corresponding cells that innervate and change their transcriptome dependently. So that I think is also very important. If you have a tumor, how can that maybe even already affect the metastatic niche, for example, or other organs in your body, also coming then related to stress and symptoms of cancer that you have, that we barely link to neural innervation. But I think it's really cool now going more and more into this field, linking other symptoms that you experience also to these innervation patterns.
That's fascinating. I think that's really fascinating. I wonder also, Vera, if you could speak to this idea of, you were talking about the sort of the tumor communicating in, the innervating neuron. I just wonder at what point does the reprogramming become clearly different and hijacked? I mean, is it one tumor cell, 10 tumor cells? And can your trace and seek be used to detect possibly an aberrant program, an aberrant neural program prior to the real clear clinical identification of the tumor?
This is an excellent question and something that we, of course, also thought about. So, what we're in general already doing is like tracing different stages of tumor progression. And I think we have some data of when the tumor is still very small and the changes were, let's say, a bit less, although it was already interesting how strong they were. I mean, this reprogram seems to happen fast. We are still tackling the problems of like how start, for example, with when there's just a pan-in, like this pre-malignant regions, because of course we need an area to inject the dye to very specifically only treat this area. Of course, then an already bigger tumor gives us a lot of area to inject the dye. When we just want to have specifically this region, that makes it, of course, much more trickier to start with. So, I mean, there you need a bit more of reporter mice and so on to really identify where this specific structure is, tackle it early on. But of course, this is also experiments still ongoing because, like everything else in this field, it's method-wise difficult to tackle, but also reaching to interesting results in the end. So we're definitely on that.
It's fascinating stuff. I look forward to maybe hearing more about that at the Cancer Neuroscience Symposium. I wanted to get back because it just fascinates me, this idea of memory. And so, say, for example, you completely cure the tumor, the pancreatic tumor. Is there still a memory in the neural network, or maybe in the neuroimmune network, of that prior experience? And does it erode with time? Does it remain fixed? Could you speak a little bit more to this idea of memory that you mentioned earlier?
I think that it has a memory, like I mentioned earlier. I mean, we could only show that when we resected the tumor, which is, of course, a different form of curing it.
Right.
If, like, for example, a treatment that actually then diminishes the tumor would induce the same memory, I don't know, because, of course, often also treatments affects the neuronal structures themselves. I mean, we could show that, for example, taxanes directly affect neuronal structures. So there, of course, that we might find differences. But in terms of memory, I think that some changes maintain. I think they will probably fade over time. I mean, we did it, of course, in mice experiments, so we can only do experiments in a, specific setting of time. In humans, of course, sometimes you have a relapse after 10 years. That would be, of course, incredibly interesting to study. With mouse models, I think this will be a bit more difficult. So we saw a reduction of the transcriptomic signature over the time, and specifically the sensory one was going down more compared to the sympathetic one. So the sympathetic ones seem to be a bit more fixed, like they are also the intense drivers of tumor progression. So I think it might fade, but I also think that some of them will remain. And this can also, presumably in humans after 10 years, at least aid in relapses.
I
Yeah,
think
yeah.
like and I want to challenge this a little bit farther because the beauty of the neurons is that they're not part of the microenvironment. Like when you're cutting the tumor out, the soma is still out there, and like we heard from Andreas, it's really hard to like reprogram cells, and it's pretty rare occasion. So they're being hijacked and then being hijacked back. With time, I feel that there are still, facets of the tumor or cancerous reprogramming that are remaining, and unlike other conduit of dissemination, like cancer doesn't have a lot of hubs that where it can, send its information. It's either local invasion, lymphatic, and also hematogenous spread. But with nerves, you actually transfer information as well. So I think that once this information has been, distributed outside of the tumor, there's a lot of things that happening not only at the first order neuron, but maybe in the secondary, maybe in the CNS, and those things usually tend to last. I'm kind of curious from this functional perspective, Vera and Andreas. Do you have any plans to looking into it? We were talking about reprogramming mostly at the transcription level and maybe at the secretome and maybe on the surface, surfaceome. But what about the electrical activity? What about the neural transmission, which is kind of the bread and butter of the neural communication, the neural function?
It's a
That
very
is
interesting question, Moran, and it burns down to the question how much of the tumor cells signaling is required to maintain the transformed or the reprogrammed neuronal state. We know that there is a memory, but we don't know actually how long that exists. So if the tumor cells disappear or taking out and there are no longer tumor cells around, I would bet that this memory state would actually return to some degree back to normal because obviously the normal cells have also interest in actually having the neurons functioning and also in this sort of, let's say, surgically operated pancreas cells. Actually, there is again a Darwinistic evolution to actually get the normal going again. But I would doubt that actually this reprogramming is totally returned to back to the stage beforehand. And that actually would also explain that some of the side effects and the misfunction of the pancreas actually is retained, even that there are no tumor cells left anymore, because US surgeons have completely removed the tumor. And well, it happened in some case, and and we also know that in for multiple myeloma or actually also in AML that actually some of the changes of the normal blood cells are actually retained, even that the tumor cells actually have been completely eradicated. So you will end up in a sort of like a hybrid state between the tumor reprogrammed one and the original wild type one. That, however, is a pure hypothesis, which actually we need to set up experiments to actually demonstrate.
I think one of the reasons that it's hard to answer this question is we're not quite sure just yet what are the causes for the reprogramming. Because once we figure out what are those factors, then we know if they still exist when the tumor is gone. If the neurons or the tumor innervating neurons are dependent on continuous signaling of something, but until we figure it out, what are the drivers for this reprogramming, it's going to be very hard to answer this question.
fully agree with you. Well, I mean, to be honest, it's of course always great to look already at the very broader picture and of course also going with the study and so on. So of course it drives me and gives me motivation to think that it will extend also over years. I think at the moment it's still in such an early state that this model that we now developed or like this data set we developed is the first of its kind. So of course, in the first next step is of course now actually applying it to different cancer entities to see if we see similar changes, if we get a comprehensive mapping of the neuronal landscape of both healthy and tumor innervating neurons. Because I think going into human, you can of course only do when you now have a very specific question, as getting material from human is so much more difficult. Of course, you can get, for example, healthy cells from donors, and you can try to get it, get also tissue from cancer patient who sadly then would have died and get material. But of course, you only want to get into that very precious sense after you have really a very good idea of what you want to look at. And I think this is currently ongoing now. We're really expanding this. And the beauty is that it doesn't even have to be limited to just cancer or metastasis. It can actually be applied also for so many other diseases, as we showed also for pancreatitis, to also compare if like changes in a disease are like similar in a neuronal content, because of course it's still innervating the same organ. If we find even similarities between different disease states. and then really going in more into human tissue. Of course, I think with the help of surgeons and so on, you can already start putting that into a context, of course, with spatial to see, hey, I see this factor changed in a neuron. Is this corresponding receptor, for example, also deregulated in the interacting cell? So I think we can already start it both ways, but as it is such a young field, I also think it still needs a bit more even on the basic research. So coming in from two ways, basically.
I think there are so many questions in so little time, but this is part of why we have these conferences, and we're looking forward to continuing this discussion at the Cancer Neuroscience Symposium and at MD Anderson in February. So I want to just take this moment to thank you, Vera and Andreas, for participating in this chat, along with you, Moran and Kenzie and Allison and Shannon. And we very much look forward to your presentations. We're learning more about the broad applications of Trace and Seek and other tools that you may be developing in the pipeline. And all in all, just wonderful conversation. Moran,
Yeah.
some final thoughts too?
Thank you. First time I've met Vera was in one of those conferences, and it actually created, a beautiful collaboration. We've been working and communicating since, and that's one of the beautiful thing about our community. So I'm just curious, what are we going to hear from you guys in the next Cancer Neuroscience meeting in 2026?
We, of
Perfect.
course, also very much looking forward to this. And also, I can just say that I'm also really happy that I dived into the field of cancer neuroscience, because I think it's really cool when you join early on and really see the field develop and how much more recognition it also gets over time. And I think it's really a very young, but also very rewarding field where everyone is really nice. And I think for me, that was also just a great experience that when I started as a PhD and we met on the first conferences, that actually you were taken seriously, you were asked for your advice, and everyone is extremely helpful. And I think that is also the beauty of this field.
Excellent. Excellent. Well, thank you both, and we look forward to seeing you again. Thanks for tuning in today. We hope you'll join us as we continue the conversation in Houston and virtually at the 2026 Cancer Neuroscience Symposium hosted by MD Anderson and Wiley's Advanced Portfolio.